Hello everyone. So today we with you on the presentation about cell culture. I have that in my lab. So what is exactly cell culture? Cell culture refers to the laboratory methods that enable the growth of eukaryotic or prokaryotic cells in physiological conditions, and is introduced to study tissue growth and maturation, virus biology, and vaccine development, the role of genes in disease and health. And in our lab, I have nurtured the Haga cell for 2 times, 2 or 3, I don't remember exactly. And I have seen the other member in our club also nurture the melanoma and see how it was like they treat with the different drug, and they see that the drug can cure or treat this disease or not. Yeah, the heart the heart trends that I'm very happy to see the techniques. I'm very lucky at all that yeah, I can see many techniques in our lab that he said that, oh, you're very lucky. Yeah, so come back to ourselves, like we have come back to our PowerPoints, that we have a lot of not a lot, like I just have 3 main cell culture named primary cell culture, secondary cell culture, and cell line. But I just want to say more about the primary cell culture. I'm not sure the Haga cell is adherent cell. Adherent cell means like the cell will bind to the bottom of the blade, and suspension cell like it not bound to, like it contrastly it not bound to the bottom of the cell. And how can you know it? It have the website name ATCC. Yeah, and you can search the name of the cell, but yeah, it's not that it don't have that kind of cell that we nurture. But for example, you can search with like blood cell in here. It doesn't sound like it's just like the store, but for example the shop, but it don't have any kind of set that you want. So yeah, so but it will have a lot of like common cells. For example, when you search with blood cell, for example, when you search that, it will have the um medium. It will tell you the medium, that how can you can nurture a cell, the product category, and yeah, you can see the information about the cell on this website also. And come back. So Haga cell is a adherent cell. Why we must know that? Because some technique like MTTS say if it's a suspension cell, you must use some chemical to make a suspension cell like bind onto the bottom. It make you easier to, it make you can treat the absorbance of this. So I don't want to tell more about this because you can search it easier into the on the internet. So I just want to share some tips that I nurture the cell and the protocol in our lab. So the protocol, first we have the Haga cell from the freezer, and we defrost it by incubate 37 degree 5 minutes with a specific medium, and we see the cell through microscopy to make sure that the cell live and the cell live. And sorry, this is step 2, and we absorb the ambient medium by vacuuming, and we added 100 microliter BBS to wash, and then vacuum BBS fluid by vacuuming. Why we must add BBS? BBS is the important chemical because it can maintain the osmosis of the media. When the, for example, like when the cell grow up, it will secrete a lot of dirty things, and PPS can help to maintain the pH of this medium. Like you can search like the cell needs the like the pH that is from 7 to to 7.4 more acids. So and after that we add 100 microliter trypsin and incubate for 37 degree in 10 minutes while we might add trypsin. Trypsin is the chemical that it detect the monolayer with the adherent cell, like bind to it, it will detect it. And step 7, like we add 1000 microliter DMBM to stop solution. Why? Because DMBM have LBS. And for the step 8 is suspend fluid and then centrifugate 1500 rate for 5 minutes. Then we vacuum the fluid except the cell and add 1000 microliter DMBM. And for the final, we will get 200 microliter fluid and add it into 4000 DMBM fluid, and we incubate 37. And the process of natural cell line, after every 3 days we will do this procedure once. So yeah, this will lead to the end of my presentation about cell culture. Thank you very much for your listening.